Hello everyone, and welcome to our unit discussing the cardiovascular system. We're going to begin by talking through Lecture 12, which covers our liquid connective tissue, blood, and the pumping mechanism that circulates blood, which of course is the heart. We're going to start first by examining blood itself and the components that make it up. Now, blood is the connective tissue that unites the entire cardiovascular system as well as most of the other organ systems in the body. Its functions are varied. The obvious one is transportation. It moves things like gases and nutrients around the body. It also participates in regulation, helps regulate the body's pH and also the body's temperature by distributing heat or helping conserve it. And it acts as a protective substance. Not the blood itself, but certain components of blood are active in hunting down and destroying microbes, collecting toxins and removing them from the body, and also to help prevent dehydration. Blood overall has a slightly basic pH. That's just a characteristic you need to know. It's a mix of a fluid called plasma, which you see here. It's this pale, semi-translucent material. And floating in that plasma are a mix of different cells, which we call formed elements, and also other components like blood proteins. We're going to talk through all of these different substances, the formed elements, which are red and white blood cells, as well as these things called platelets, and also proteins, nutrients, dissolved gases, basically everything else that can be transported by blood. All right, here's an overview of the composition of plasma. Now we're going to talk through plasma a little bit on this slide, but this is your orientation. You need to know this basic information about the proteins in blood. They're also discussed on the next slide. And you just need to know that these other solutes, remember a solute is something dissolved in a liquid or a solvent, you just need to know that these are present in various amounts. You also need to know that plasma is primarily water. Okay. You'll see that on the next slide. All right. Remember that plasma is a fluid matrix. Remember, blood's a connective tissue, right? So it's living cells suspended in a non-living matrix. The plasma is the non-living matrix. It's 92% water, but that amount varies with your hydration. If you're dehydrated, that number is going to be lower. It transports all of those materials we talked about before, but there's a few that I want to focus on. These are called plasma proteins. Albumin is by far the most common protein in the plasma. It makes up 60% of all proteins found in the blood. And albumin is important for a lot of reasons, but one of the biggest ones is it contributes to osmoregulation. Now, osmoregulation is exactly what it sounds like. Think of osmo as in osmosis. This is going to have something to do with water. Regulation is control or management. Albumin actually helps control the water balance in your blood. Now, blood flows through your blood vessels at high pressure. Remember, water molecules are fairly small. They can actually be pushed out of the blood vessels by the immense pressure and into the surrounding tissues. This leads to a condition called edema. Edema. There we go. Edema is swelling, localized swelling due to a release of water into surrounding tissues. Now, what albumin does is it actually helps to block the water from being able to escape the blood vessels, concealing it inside and preventing edema. There are a couple other plasma proteins you should know about. There's something called globulin. There are alpha and beta types of globulin. Just know that those two forms exist. Globulin is responsible for transporting lipids, or fats, and also vitamins. There is also a gamma type of globulin. The gamma globulins are usually just referred to as immunoglobulins because they're globulins that participate in your immune response. Their common name, antibodies. Antibodies are immunoglobulins. So we're going to talk about antibodies more later. Finally, last protein, and my favorite, fibrinogen, pardon me. Fibrinogen only makes up about 4% of the proteins in the blood, but it's critically important. it participates in blood clotting, which is a really fascinating process. Now, what determines the volume of plasma you have in your body? How much blood plasma does your body really need, and what makes that determination? Well, there are these nerves in the hypothalamus region of your brain called osmoreceptors. So these are water detectors. Okay? They detect when your plasma level is too low. Okay? This is a state we call dehydration. They'll then send a signal in the form of a chemical called ADH. It's the primary hormone responsible for controlling water retention and thirst. When ADH levels are high in your blood, you feel thirsty and your body tends to retain more water. This is to recover from a dehydrated state. So if you've ever had that experience where you're really thirsty, you start drinking a glass of water and you feel like you can't stop, that's probably because of an excess of ADH in your body. Your body's desperate for water. All right, that's it for plasma. Now we're going to move on to the formed elements that are found in blood. A formed element is something that's a solid or a molecule. Okay? The majority of the formed elements that we are going to discuss are cell types. You can see them here. Platelets, white blood cells, and red blood cells. And looking at these numbers, it should be pretty apparent that the majority of cells in your body are going to be these RBCs, or red blood cells. Platelets are pretty distinctive in their shape. They look like cell fragments because that's basically what they are. White blood cells have the greatest diversity of cell shapes and types. Red blood cells are pretty homogeneous in appearance. They all look like little red donuts that don't have a full hole in the middle, kind of like lozenges. We'll start by talking about those red blood cells. They are formally termed erythrocytes. Erythro meaning red, cyte meaning cell. Okay? We also tend to call them RBCs for short. Red blood cells are by far the most numerous formed element in the blood. They also lack nuclei, which is very, very distinctive. They only have a lifespan of about 120 days before the body recycles them and replaces them. And probably most important, they contain a molecule called hemoglobin. Hemoglobin. This is a globulin. In case you missed that, the globin part gives it away. Hemo means blood. It's actually a reference to this molecule over here called heme. Okay, so there's a heme molecule. If you want to see what the actual molecular structure looks like, it's down here. It's very complex, but what's critical is this little guy in the middle. That is iron. And iron has a wonderfully high affinity for oxygen. Okay? Hemoglobin itself, it's a complex of 4 proteins. They're listed here and called chains, chain 1, 2, and then chain 1 and 2 over here refer to an alpha and a beta strain of proteins. Just know that each of these chains, alpha 1, beta 2, beta 1, alpha 2, each of those is a protein complex. So there's 4 total. Each of these complexes is bound to its own heme molecule. Okay. The iron in these molecules is eventually recycled. So you know that these blood cells only live 120 days. The iron, the 4 pieces found in each heme molecule or hemoglobin molecule that they're carrying, that iron isn't lost when the blood cells are broken down and recycled. It's reused over and over. And the liver is what's responsible for capturing it and reusing it. All right, I alluded to this before, but let's spell it out. Hemoglobin is really important because it's responsible for both the collection and the release of oxygen. We're going to talk through these mechanisms in a later lecture. For now, know that hemoglobin is really good at binding to oxygen when oxygen concentrations are high. So if you put hemoglobin in a high oxygen environment, it sucks up that oxygen. But if you take that loaded up hemoglobin and then stick it in an environment where oxygen is low, any oxygen the hemoglobin is carrying, it will release to try to bring up the oxygen concentration in that low environment. This makes it the perfect mechanism for transporting oxygen through the body. It'll pick up oxygen in the oxygen-rich environment of the lungs, and then as it travels through the bloodstream in the body, as it moves into your muscles or into your organs, which are low oxygen environments, it will release that oxygen as it goes, oxygenating your systems and allowing cellular respiration to occur in your cells. And again, that iron eventually gets recycled in the liver. We don't waste it. Now, this is kind of cool. A single erythrocyte can contain 300 million hemoglobin molecules. Remember, each hemoglobin carries 4 heme molecules. If you do the math on this, it means more than 1 billion oxygen molecules can be transported by one erythrocyte. So these cells, the erythrocytes, not just the hemoglobin, but the erythrocytes are really, really good at picking up oxygen and transporting huge amounts of it. All right, moving on to our white blood cells. These are called leukocytes. There are fewer in number. There's only about 1 or 2 for every 1,000 erythrocytes. Their job isn't to transport materials. Instead, these are the protective cells in blood. They function in immune defense, and they use phagocytosis to engulf and break down things like microbes and toxins, removing them from the bloodstream. They're significantly larger. You can kind of get a sense of that from this image. Here's a red blood cell. This giant structure is your white blood cell. You can see it down here in these slides as well. Here are your RBCs. Here's your white blood cell. The other big difference is these guys have nuclei, and you can see them in these slide images. These dark blotches in the middle of each of these cells are the nuclei. The other thing that's different between leukocytes and erythrocytes is that leukocytes aren't confined to the blood. They can actually migrate out of blood vessels. This is referred to as, I'm going to change my color here real quick, if I can. Yep. Change it to our normal highlighting color. This is called diapedesis or diapedesis. Okay, this is a word you need to know. I'm sorry it's not already highlighted. Okay. This is the act of a cell leaving a blood vessel, being able to move through a membrane into other parts of the body. Leukocytes patrol the surrounding interstitial fluid, meaning the fluid that exists between organs, between blood vessels, or they can move straight into what's called the lymphatic system, which is another fluid distribution system that we'll talk about in a later lecture. In essence, leukocytes can travel just about anywhere because microbes can enter your system in a variety of different places. And so you need white blood cells present in a lot of areas to keep watch for infection. Now there's a lot of different types of leukocytes. You saw that on the slide, the overview slide about 3 slides ago. We break them down into 2 larger groups. We call them either granular leukocytes or agranular. We'll show you those on the next slide. Granular leukocytes are called that because they have what look like granules or little grains in their cytoplasm. The cytoplasm literally looks grainy or textured. They also have lobed nuclei, meaning the nuclei have multiple round protrusions on them. There's 3 types you need to know about. The eosinophils, which are right here. Okay. These guys are very granular. You see all that texture there in the background? Those are the grains. They turn pink with an eosin stain. So if you were testing these in a lab and you were to apply that stain, any cells that are obviously larger than a red blood cell and that turn pink with the stain, that's an eosinophil. Basophils are granular as well. Again, you see all these grains in the cytoplasm. They turn blue with just a basic cellular stain. Eosinophils won't, so that's another way to rule them out. Okay. Neutrophils, interestingly, have no affinity for either stain. They will be somewhat pale in color in the background. Their nuclei will die, but the rest of the cell will remain mostly transparent. Okay, so it's just another way of sorting out cells with a stain. And again, you see all the little dots? This is how you know it's a granular leukocyte. Each of these leukocytes has different activity in the immune system, which we'll discuss in very broad terms later. For now, I'm just introducing you to the cell types. All right, so those are your granular leukocytes. Now let's look at agranular. Agranular leukocytes, first, where's my pen at? There it is. They have a relatively smooth looking cytoplasm. There aren't obvious dots or grains like what you see in granular leukocytes. They also have large semi-rounded nuclei that look like they're a single lobe, okay, or maybe two really big lobes. They're not nearly as complex in appearance as the nuclei in your granular leukocytes. Okay. Lymphocytes and monocytes are the two agranular leukocytes you need to know about. Okay. Notice too how massive the nuclei are on these agranular leukocytes when you compare them to the granular ones. Nuclei size is another giveaway. All right, now on to my favorite group, the thrombocytes. These are called platelets. Now keep in mind, platelet is the common name. Just like we would call a erythrocyte a red blood cell, and red blood cell is its common name, platelet is the common name for this cell type. Thrombocyte is its formal name. On this picture, the thrombocyte is actually the source of these long thread-like materials. So all of this stuff that I'm highlighting, all of this is being created and generated by thrombocytes. Okay. Thrombocytes are actually pretty much just cell fragments. They pinch off from larger cells during cellular genesis in blood marrow. They also participate in regulation. Their job is to stop blood loss in the occasion of in case of injury. So following injury, platelets release something called a clotting factor. Clotting factors are proteins. They're carried by the plasma in the blood, and they flow around, and their job is to go around and activate other platelets, calling them to the site of the injury. So clotting factors attract more and more platelets to the site of the damage. Okay. All right. Here's how clotting works. Okay. When platelets contact collagen, they stick to it, right? So platelets and collagen have an affinity for each other. Now imagine this is the cross section of a blood vessel. Okay. So flowing through the inside of that blood vessel, there are, hold on, all sorts of different blood cells, plasma, and mixed into all of that are also platelets. Now, another thing to keep in mind is, remember that connective tissue often contains large amounts of collagen, which I'm going to represent as blue. Okay? So out here, this cell is surrounded by collagen fibers, or I'm sorry, not cell, this blood vessel is surrounded by collagen fibers, anchoring it in place. It's also surrounded by a layer of connective tissue made of collagen. So you get the idea. Blue is collagen. There's a shitload of collagen surrounding this vessel. Now, if we were to create a hole in that blood vessel, options, we can erase a tiny bit. Oops. Well, actually, that'll work. Gives you a little bit more clarity. Let me change my ink color one more time. Change it to red. All right, so imagine damage is done to this blood vessel. and you start having blood rupture out of the vessel and into this space. Collagen is going to be in direct contact with this blood, and in the blood there are platelets. So platelets don't encounter collagen when they're safely inside the blood vessel. They're separated from it. But once the blood vessel wall is broken and blood starts leaking out, suddenly those platelets are exposed to the collagen that surrounds the vessel, and they start to stick to it. Exposure to collagen also triggers a chemical cascade inside the platelet. It causes a conversion in the platelet of fibrinogen, which is a soluble protein, into a different protein called fibrin, which is insoluble. Now remember, plasma is primarily water. Platelets usually carry around fibrinogen. This stuff just dissolves in water, doesn't hold up the flow of blood at all. But when this fibrinogen is changed to fibrin, which is insoluble, it starts taking on solid form inside the blood. Suddenly, it's going to interfere with blood flow. But it's only going to do this wherever platelets are in contact with collagen, which is right here at the site of the injury. All right, so on this diagram, here's where we're at. So we have an injury made to our blood vessel. Red blood cells and platelets are what are displayed here in this diagram. What it's showing you here is what the platelets do when they contact the collagen on the outer surface of this blood vessel. They stick to it, then they stick to each other, and they form a plug. Okay? Those cascading reactions I talked about are going to turn your fibrinogen into fibrin. And that fibrin is going to create threads. These threads are going to grow and grow and then link together, forming a net, which is what you see here. All of these lines are lines of fibrin. Those fibrin lines are going to attach to each other, which means they're going to connect adjacent platelets to one another, and they're going to create what's almost like a fishing net. Any larger cells that try to leave through the hole in the blood vessel, through the injury site, they're going to get caught in that net and trapped there. That's what you saw on this slide. This is a red blood cell that's been captured by this fibrin net and held in place. Platelets, thrombocytes, actually create a physical plug in any hole in a blood vessel by using their own bodies and any captured cellular bodies that they catch in that fibrin net. This combination of platelets and other cells and fibrin, that's what we call a blood clot. Okay? If healing occurs naturally, the cells that make up the walls of the blood vessel will eventually knit themselves back together. That will cut the platelets off from the collagen on the outer surface of the vessel. When the platelets are no longer in contact with that collagen, they stop producing fibrin, and eventually all of the components of the clot, the pieces of fibrin, the captured cells, the old platelets, will be slowly broken down and washed away a little at a time back into the bloodstream, where they'll then be captured and filtered out. It's a pretty cool process. All right, so we've talked about our formed elements. Now we need to talk about where they come from. The process of blood cell formation is called hematopoiesis. Think heme, hema, think of this prefix as meaning blood. What triggers blood cell production is a hormone called erythropoietin. Okay? Erythropoietin. This is produced when your blood volume when your blood cell volume gets too low. If you develop anemia, you have too few red blood cells, your body will start overproducing erythropoietin to try to bring that number back up. There is actually only one stem cell that gives rise to all of our blood cell types. It's called a hemocytoblast. This is a stem cell for all blood cell types. You can take a hemocytoblast and eventually mature it into either an erythrocyte or a leukocyte or a platelet. The trick is it can only develop into one type. It can't then reverse and change back into something else. During embryonic development, when your blood is first developing, hematopoiesis occurs in a lot of places. It happens in your bone marrow, in your liver, in your spleen, happens in your thymus, it even happens inside your own blood vessels. But that changes after birth. Once you're born and mature into an adult, hematopoiesis is confined primarily to red bone marrow. Yellow bone marrow can do a little bit of it in emergency circumstances, but the vast majority of your blood cells come from your red bone marrow. This is the process of hematopoiesis. I don't want you to spend too much time on it. I just want you to get a sense of the general flow. So we start inside the marrow cavity of a long bone. We focus in here on the red bone marrow, and that's where we find our blood stem cells. They can take a couple of different developmental routes. I don't want you to worry about these intermediate steps of myeloid versus lymphatic stem cells. Just note the variety of different cells that can develop from this one stem cell. It can give rise to, if you follow this line, all of your red blood cells. It can give rise to your platelets. It can give rise to all of your different types of white blood cells as well. Don't worry about the intervening steps. Okay. All right, now that we've got a foundation in what blood is made of, let's talk about how blood testing works, what you might be doing in your clinical practice. Probably the most basic, but also most common test you may need to do is called a hematocrit test. This is where you measure the ratio of the volume of red blood cells to the total volume of the blood. So you're comparing how many red blood cells there are in a certain volume of blood. Okay. The way you do this is you take a blood sample in a tube, maybe one that looks kind of like this guy. Okay. And when it starts out, it's a completely mixed tube of blood, so it's solid red. Okay. You stick this tube inside of this machine, which is called a centrifuge. You put the tube in at a slight angle, and then you spin the hell out of it, over and over and over again. This is like the ride at the fair where everybody goes inside a big cylinder. You stand up against the wall and they start to spin it, and you stick to the wall and kind of get lifted off the ground. That's what it feels like inside a centrifuge, only even stronger. The effect of this spinning is that the denser particles in the blood are pulled to the far end of the tube. Oops, where is there it is. Down here. So all the denser particles get forced downwards. And they form a mass. Kind of looks like this. This is where all your cells are. Primarily, of course, your red blood cells, because that's what most of the cells in your blood are. Okay. What you do then is you compare the total volume of fluid inside that tube, the total volume of both cells and plasma, and you compare that overall volume to the size of this mass of red blood cells. Okay. We call that the hard fraction because it's made mostly of solids, so this down here is the hard fraction. The height of the hard fraction, which is literally measured using a ruler, you divide that by the height of the entire sample, liquid and all, and you multiply it by 100 and it gives you a percent. So in this example, the hard fraction goes up to about the 4.7 centimeter mark. Total volume is up here at about 9 centimeters even. Divide those, multiply by 100, and what you get is the number 52. That means 52% of this blood sample is made up of erythrocytes, or red blood cells. All right, so how do you interpret these results? What does that number actually mean? Well, first you're given baselines. For males, the normal hematocrit levels are 37 to 47%. That means 37 to 47% of their blood should be made up of RBCs. For females, it's a different number. It's higher. Females should have more red blood cells, between 42 and 52%. Okay. Remember that what you have done is you've separated your blood into plasma into red blood cells, and if you were to look close, you would notice there'd be this little white line in between. Real thin little white line. That's called the buffy coat. It's made up of white blood cells and platelets, your other formed elements. They aren't as dense as red blood cells, so they don't get pulled down as far. Now, knowing this is what normal looks like, let's look at some abnormal results. All right, so here we have a whole lot of plasma. We've got a normal seeming amount of buffy coat, and we don't have very much erythrocyte. We don't have much of a solid or of a solid layer. This indicates your hematocrit is too low. We call it a depressed hematocrit. You don't have enough red blood cells. This is called anemia. When you are anemic, your body is usually somewhat oxygen starved. Common symptoms are becoming very tired, trouble thinking, irritability, and also your nail beds start to turn blue. And that's because of a lack of the red pigmentation that comes from hemoglobin, because you just don't have that many red blood cells to turn those nail beds nice and red. And you also are low on oxygen, which makes hemoglobin appear even more red. Now, the opposite can happen too. So if we look over here, suddenly we have a huge amount of red blood cells. This is polycythemia. This is elevated hematocrit. You've got too many red blood cells. Okay, so both conditions can occur. All right, after hematocrit testing, blood typing is probably the second most common blood test you might experience. First thing to know is that there are different kinds of red blood cells. In fact, there are 4 different types: OO, AO, BO, and AB. It's like alphabet soup. Each of these letters refers to a gene that you inherited from either your mother or your father. Okay, so this is a representation of 2 different versions of a gene. These 2 different genes actually affect the structure somewhat of your red blood cells. Let me show you how this works. The way that we determine your blood type is we test for certain antigens and whether or not they're present on your red blood cells. That's actually what you wind up inheriting from your parents. So what are antigens, and what are the things we use to test for them? Antibodies? Well, ooh, that's out of order. Hang on. There we go. Put it all up. Okay, antigens, often abbreviated to just AG. The word antigen itself is actually an abbreviation. The anti comes from the word antibody. The gen comes from the word generator. Antigens are antibody generators. They're molecules that act as identification markers on the surfaces of all cells. But the trick is, since they're an identification marker, they can flag a cell as being foreign to your body. And thus, these antigens can stimulate an immune response if that cell is in a place it's not supposed to be. Okay? We say that antigens are either self or non-self. That means your body either recognizes antigens as belonging to you, they're yours, or it doesn't recognize them and tags them as being not yours. Your body's default reaction to encountering cells that don't belong to it is to try to destroy them. They're probably dangerous. Now, these antigens are what are actually genetically inherited. You get your specific antigens from your parents, your mother and your father. The way that we detect antigens is by using those antibodies that they are so good at triggering the production of. Okay? Remember, antibodies are proteins. They react with foreign antigens, grabbing onto them. So you can see them down here. The green are the antibodies, red are the pathogen, which is also the antigen. The antibodies literally grab on to these pathogen cells, creating this lump, this grouping. It's a kind of clotting that we call agglutination. Agglutination means the glopping together or the glooping together, if you want to use nice alliterative words, or the clumping of antibodies and the cells that they are reacting to that are covered with their antigens. This clumping makes the agglutinated antibody and cell complex really obvious. It's hard hard to miss something this big floating through the bloodstream, which means it makes it really easy for the immune system to identify this structure and to destroy it. All right, here's how this works. If you want to determine the blood type of someone, you have to determine what type of antibodies they carry on their blood cells. There are two different what kind of antigens they carry on their blood cells. There's two different antigen types. We use the letter A to represent one. We use the letter B to represent the other. Okay, so these are your two different types of antigens. Now, what you do is you take blood samples from a patient, okay, and then you mix them with antibodies from each blood type group. Okay. So, if your patient has, for example, type A blood, okay, it's going to react just fine with the antibodies from another individual with type A blood. No problem. Type A antigens will always match with type A antigens. There'll be no reaction. See how there's no clotting here? The cells are just free. That's an indication that your patient has type A blood. You mix it with type A antibodies, no reaction. That's because it's type A. But if you mix type A antibodies with, say, type B blood down here, so this is type B blood from your patient, you mixed it with type A antibodies, which is what's up here along the top. See how there's clustering, clumping? Okay. That's agglutination. This is an indication that your patient does not have type A blood. Okay. Now, check out this down here. You've mixed type A antibodies with your patient's blood, and there's no clotting. None at all. Now, this could mean your patient has type A blood. But really, all it really means is your patient has type A antigens on at least some of its blood cells. It has type A antigens on its blood cells. That doesn't mean that's the only antigen it has on its blood cells. Here's how this works. All right, let's go to type B antigens. If you mix type B antigens with type A blood down here, probably understandably, you're going to get agglutination. Type B antibodies are going to react with type A antigens. Now, if you mix type B antibodies with type B antigens, you'll get no response because they recognize each other as being like. Down here, mixing your patient's blood with type B antibodies also creates no response. Now, this is a little weird because this patient down here on the bottom row, we also got no response when we mixed their blood with type A antibodies. No response here, no response here. What this should immediately tell you is that your blood cells in this patient have both A and B antigens. That's why the antibodies don't react with it in either case. Both type of antigens are present, so the antibodies recognize them and leave the cells alone. If you were to mix your A and B antibodies together, those A and B antibodies would react badly with your type A blood because the B antibodies would attack the cells. It would also react badly with your type A blood up here. Also react badly with your type B blood because the A antibodies would attack those B cells. But if you mix A and B antibodies together and expose them to this last patient, again, there's no reaction. Because again, there are both A and B antigens in this blood that are recognized by both your A and B antibodies. All right. There is one more situation, one more case that we have to discuss. It has to do with this O type of blood. A is an antigen, so is B. These are markers on the outsides of cells. If you have type O blood, you lack either of those markers. You don't have any markers, no antigens on your blood cells, nothing for the antibodies to react to. If you have type O blood and you're exposed to type O antibodies, which we can manufacture, there will be no reaction. That makes sense. They're the same thing. But check this out. What happens when you mix type O antibodies with type A blood? Nothing. It's because these O antibodies don't react with anything on that type A blood. It's a null result. Same thing with B, same thing with AB. Now, if you look at your type O individual, so our individual who has type O blood, notice they don't react with the O antibodies, but they react badly with every other kind. The A antibodies are looking for A antigens. Type O blood doesn't have any antigens. That's going to register as a not self reaction, and there's going to be agglutination. B antibodies are looking for B antigens. There aren't any on O blood. Again, agglutination. Expose it to both A and B, you're going to get the same result. Okay? Any individual, regardless of blood type, can accept type O blood because there's no antigens there to trigger a negative immune response. Type O blood is the universal donor type. Anybody can receive it. Okay? Type AB blood, if you notice, hasn't reacted to any of our antibody tests. Didn't react to type O antibodies or A or B or AB. Type AB blood is considered to be the universal acceptor, universal receiver. Individuals who are type AB can receive any kind of blood. The opposite end of that spectrum are people with type O. While people with type O can donate to anyone, they can only receive from other people with type O blood. So why is there never agglutination with type O? There aren't antigens to trigger it. And that statement, why is there never agglutination, it's referring to this first column. All right, it's probably obvious, but the reason blood typing is so important, so critically important, is when you're talking about blood donation. If you give a patient the incorrect blood type, they're going to experience rampant agglutination, which is basically blood clotting, and it can interrupt blood flow, sometimes unto death. So the earliest experimentation with blood transfusions in the 18 and 1900s oftentimes killed the patient because they were transferring blood from an individual with an incompatible blood type into the patient. Just made things worse. Now, there's actually a second aspect of blood typing other than their antigens. Blood cells also have what's called an Rh factor. That's an abbreviation of rhesus factor. The Rh factor is also genetically inherited, just like your antigens. It's a protein, and it's found on the outside of erythrocytes. It will also trigger an antibody response. If an Rh negative patient, meaning an individual who doesn't have these proteins, receives Rh positive blood, meaning they get blood that does have these proteins, their body will recognize the proteins on the new blood cells as being not self and attack them. The opposite's also true. So this chart down here is a nice little comparison showing you compatibility and incompatibility between recipients and donors of different types of blood. Notice that O negative is the true universal donor. O negative as a donor can give blood to everybody, but O negative as a recipient, it can only receive blood from somebody else that's O negative. Right? Down here, interestingly, it's kind of the opposite. AB positive is the universal receiver, universal recipient. It can get blood from anybody. Okay? All right. One thing about Rh factor that's interesting is a condition that can develop as a result of pregnancy. It's called Rh sensitization. Rh sensitization, if it occurs, it can cause pregnancy complications, resulting in the need for close monitoring of the fetus, even in extreme circumstances, a fetal blood transfusion. And this occurs in second pregnancies most of the time. So here's how this works. If we come down here, look at our nice little diagram. Where are you at? So our female here is Rh negative. Our male, our father, is Rh positive. Okay? These two individuals conceive a child. Okay? Here's our negative female. She is pregnant. The baby, the fetus, has received the gene from the father for Rh factor, meaning the fetus has that Rh protein just like its father. It's Rh positive, even though the mother is Rh negative. Okay? It's not uncommon for fetal cells to enter into the maternal system during pregnancy. What happens is cells from the fetus can enter into the woman's body, introducing these Rh positive cells into her normally Rh negative bloodstream. The woman's body, as a response to this, starts producing antibodies. Okay? We say her body becomes sensitized. These antibodies form to fight these Rh positive blood cells. It zeroes in on them and attacks them, tries to destroy them. The woman's body now is filled with antibodies that are trained to look for Rh positive blood cells. Now, after she gives birth, this isn't really an issue. Her body isolates those Rh positive blood cells and deals with them. The problem arises if she becomes pregnant again. Okay? And if that second baby is also Rh positive. In the next Rh positive pregnancy, those maternal antibodies against the Rh factor can actually attack the fetus. Okay? And they're not just attacking any old part of the fetus, they're attacking the fetal red blood cells. If enough of them are destroyed, the fetus can die. So this is why if a mother has been diagnosed as Rh sensitive, they've got to monitor the pregnancy, make sure the baby's producing enough viable red blood cells, and in extreme circumstances, if too many of the RBCs are being destroyed, they got to do a blood transfusion, try to bring that fetus's blood volume back up. This does not happen every time an Rh negative mother gives birth to an Rh positive baby, but it is a possibility. All right, that's it for our overview of blood. Now we're going to discuss an overview of the heart. I'm not going to talk about everything on every slide because a lot of this information is, I don't know, it's pretty straightforward, pretty obvious from the diagrams and also from your lab manual, but I'm going to highlight some of the trickier bits that may need an explanation. First thing I want you to know about why this is so important, #1 killer in the United States of both men and women, but primarily women, is heart disease and heart failure. Understanding the heart is going to be a critical component in diagnosing what is one of the most common diseases or disease conditions in the U.S. Okay. So there's your plug for why this matters. Now the heart you can think of as the central pump that powers the whole cardiovascular system. Okay. The whole system is made up of the heart and all its vessels. The heart itself has 4 chambers, 2 atria, or atrium if it's singular, and 2 ventricles, or ventricle. Think of these as being in alphabetical order. A goes on top, so the atria are up here. V goes on the bottom, the ventricles are down here. Also, the ventricles have this V shape to them, which is kind of nice, pointed at the bottom. Okay. Now, there's 2 major types of blood vessels. There are arteries and veins. Arteries carry blood away from the heart. A for away. So arteries, focus on this A. A for away. That's key. Veins carry blood to the heart. Now, it doesn't matter what kind of blood it is. It doesn't matter what's in the blood. All that matters is what direction the blood is flowing through a particular vessel. So if I were to choose a tube at random and then look at the arrow next to it, so let's look at this tube, okay? Next to it is this blue arrow. It says that blood is flowing down like this. That's towards the heart. That must be a vein carrying blood towards the heart. What about this one? So we've got this tube. It starts here, and then it goes up this way. There's our arrow. It says it's going in this direction. Heart's back here. It's moving away from the heart. This must be an artery. Okay. Now, arteries and veins eventually connect to each other. They're connected by these super small, super thin vessels called capillaries. Now, if you look back at the heart, focus back at the heart is divided into two sides, a left side and a right side. Now, this is important. When you are looking at a diagram of the heart, the side that you're looking at, or the sides that you are focused on, are always based on the patient's anatomical position. Okay? So if this heart is inside the chest of your patient, this is your patient's right side. Okay? That's the right side. If this heart's in the chest of your patient, this is the patient's left side. Okay? It's the opposite of what we would think. This side may be your left side looking at the heart. Doesn't matter. It's not your heart. It belongs to the patient. So we use the patient's anatomical position. Patient's right side is here. So this is their right atrium and right ventricle. Patient's left side is here. So this is their left atrium and left ventricle. Okay. Now that's important to know because different types of blood flow through each side of the heart. Deoxygenated blood flows through the right side of the heart. Okay? This is all deoxygenated blood. Oxygenated blood flows through the left side of the heart. Okay? Now, in all animals, not just humans, but all animals that have four-chambered hearts, our circulation is divided into two different systems. So we call it double circulation. We have a double circulatory system. The first of these two circuits is called the pulmonary circuit. Pulmonary should make you think of lungs. The pulmonary circuit is blood flow to and from the lungs. The right atrium, RA is short for right atrium, it's going to receive deoxygenated blood from the body. It's going to drop it into the right ventricle and then send it out to the lungs, here and here. That blood is then going to return back to the heart and enter into the left atrium. Okay? This is the beginning, right here, of the systemic circuit. The systemic circuit sends blood to every other organ system in your body. Systemic, think of it as the rest of your systems. The left atrium is going to receive oxygenated blood. Just came from the lungs. It's going to be oxygenated. It's going to receive that blood and then deliver it down to the left ventricle. The left ventricle is then going to pump that blood up through the aorta, this giant vessel, and it'll head up towards the head and down towards the rest of the body. Okay. Oops. All right. Now the heart is an incredible organ. It's absolutely incredible. Remember, it's a muscle, cardiac muscle tissue. It pumps continuously from about a third of the way into about a sixth of the way into your development, all the way until the day you die. It never gets to rest. It never gets to stop. It never gets to take a break. Think about every other muscle in your body. You get muscle fatigue, you get lactic acid buildup. You need to give those muscles a rest. You can't walk non-stop 24 hours a day, 7 days a week for 25 years without at some point collapsing. Okay? Your skeletal muscles aren't designed for that kind of endurance. But your cardiac muscle is. It's incredible. If you're in really good shape, it'll beat about 60 times a minute. Average shape, 70-ish times a minute. 60 minutes every hour, 24 hours every day, 7 days every week. Think about the total number of beats of muscle contractions. That's incredible. The reason it has to do this is because stationary blood, if blood were just allowed to sit in your body, it would be rapidly drained of oxygen and totally saturated with CO2 and waste. The surrounding tissue would suck all the O2 out of that blood, cram it full of CO2 and waste products, and that blood would just sit there, stagnating, and the cells around it would slowly die. It has to be continuously in motion. The heart's an amazingly efficient pump, too. It pumps about 6 liters of blood every minute when it's at rest, when you're sleeping. It's 30 liters a minute when you're exercising. That's intense. That's a lot of blood flow. Now, the heart has some supportive tissue and layering you need to know about. There's the pericardium, which hopefully rings a bell from early in the semester. The pericardium is that space around the heart. It's the fibrous casing. It binds the heart to the mediastinum. The mediastinum is the bag, the area that surrounds the heart and the thymus and some other structures and separates them from the lungs. Okay? And it's bound to, in part, the diaphragm, right down here. Okay? The next layer of the pericardium is the serous layers. There are 2 of them. They're separated by a cavity filled with pericardial fluid, which is a lubricant. Okay? So the pericardial cavity is, where's my pen? There it is, right here. Okay? These are your 2 serous membranes. That's the liquid inside. So if you come over here, these would be your serous membranes. They'd be the edges of the balloon. The blue stuff would be the pericardial cavity filled with pericardial fluid. Here, the fist that's being pushed inside of it, this guy, that represents the heart. Okay. The parietal pericardium attaches to the fibrous pericardium, okay, which is down here. Right? Don't worry too much about this. I'm just sticking it in there because you may see these words in the future. I will not test you on this, right? I just want you to see it. Same thing here. Visceral pericardium, it attaches to the actual surface of the heart. This membrane on the inside, that's the visceral pericardium. This membrane on the outside, that is the parietal pericardium. Now, if we look at the cardiac tissue itself, we'll find that there are multiple layers to the heart. The epicardium, epi means above or on top of, it's down here, where's my pointer? There it is, down here, okay. It is found right up against the heart itself. Remember, this is that serous layer that covers the heart muscle itself. Then there's the myocardium. The myocardium is the actual cardiac muscle tissue you see here in the middle. Okay. The endocardium is the inner surface of the heart. This is what actually shows up inside your heart chambers. This would be the surface inside, say, the atrium. It's covered by simple squamous cells. Remember, squamous cells line the insides of cavities, and your ventricles and atria are definitely cavities. They're open spaces. Looking close at heart muscle structure, you'll see it's just muscle tissue. We've seen all this before. What I want to draw your attention to, though, is the intercalated discs. Remember, these are the things that were really distinctive, other than, say, branching, for cardiac muscle. If you look down here at intercalated disc structure, what you'll notice is this disc is actually composed of the Z-lines from the sarcomeres that are binding together across opposing cell membranes. Okay? They're reinforced by desmosomes. Desmosomes are membranous structures that hold cells together across gaps or junctions. So this is a junction between adjacent muscle cells. This desmosome is holding them together like an anchor or like glue. All right. Let's look now at the different chambers of the heart. In general, know that the atria receive blood. The right atrium receives it from the body. Left atrium receives it from the lungs. Okay. The right is receiving deoxygenated blood. Left is receiving oxygenated. Okay. Let's just put that up here. Now, the right atrium, you can't see it on this diagram, but internally it has 2 important nerve clusters. The first is called the sinoatrial node. The sinoatrial node is the pacemaker of the heart. It's what determines your heart rate and keeps it nice and regular. The atrioventricular node is actually electrically conductive. It conducts electrical impulses from the atria to the ventricles to make sure that they are beating in sync with each other. The atria contract first, followed by the ventricles. Okay? There's another structure here you should see. It's connected to the atria. This thing is called an auricle. An auricle is literally a pouch that sits on top of the heart. So this would be the auricle, this is the heart. Auricles are external pouches that attach to each atrium, and it increases their total volume. So the pouch is like extra space that blood can flow into, so the atria can hold more. Your ventricles are the chambers that are responsible for pumping blood out of the heart into another destination. The right one pumps deoxygenated blood to the lungs. Left one pumps oxygenated blood back to the body. Ventricle walls are significantly thicker than atrial walls. It's because the myocardium is thicker than in the atria, because these are the chambers that have to do the majority of the work. You need more muscle tissue to achieve that strong squeeze that allows blood to be shot out of the heart. Of all the chambers in the heart, the left ventricle has the hardest job, so it is the thickest walls in the chamber. It's got to pump blood the greatest distance. Think about it. The left ventricle down here, its job is to send blood out through the aorta to the entire rest of the body. The right ventricle just has to get blood to the lung. That's it. And the lungs are right there next to the heart. It's not a very far distance. But the left ventricle, it's responsible for getting blood all the way out of the heart, up to your head, and out to the rest of your body. It's a much greater distance. It needs a lot more muscle to shoot the blood that far. Your ventricles, internally, right in here, are separated by a wall called a septum. So septums separate chambers in the body. It's found in between the ventricles, so we call it the interventricular septum. It's also muscular, so it assists with that pumping action. All right, now let's look beyond the heart and at the vessels that are connected to it and the valves that regulate blood flow through them. Biggest blood vessel in the body, biggest one that loops off the heart too, is the aorta. Get all my arrows here. Right here. It carries oxygenated blood from the left ventricle to the body. Notice all the repetition. These are things you definitely have to know. There is also the pulmonary artery. The pulmonary artery carries blood, again, away from the heart, out to the lungs. Now let's talk about veins. Remember the aorta, it's carrying blood away from the heart, which means it has to be an artery. For veins, carrying blood back to the heart, the two biggest ones are the superior and inferior vena cava. So this is the superior vena cava up here. We don't call it superior because it's better. Remember, superior anatomically means above. This is above the right atrium. And then the inferior vena cava is down here. Okay, it's going to carry blood up behind the back of the heart and in through this little gap there into the right atrium. They're carrying deoxygenated blood back from the upper body and the lower body to the heart. Finally, we've got the pulmonary veins. They're carrying oxygenated blood from the lungs to the left atrium. So here, and then there's another set back in here that come over. Okay? So you're getting them from both lungs into this left atrium. Now, blood flow through these vessels has to be regulated. It can't be allowed to just flow all the time. The heart's a nicely controlled system, and it controls when blood can move through different areas. The atrioventricular valves are probably the most obvious. They're here. and they're what regulate blood flow from the atria to the ventricles. On your right side, you have a valve here that is made up of 3 different flaps. One flap is here, there's another flap in the back, and then this flap has been cut away so we can see. When you look at this valve from above, it's split into 3 pieces. So we call it a tricuspid valve. All 3 of these pieces open outwards like a petal on like petals on a flower, allowing blood to flow through. And then they slam shut afterwards. On the left side, over here, you have what's called just a bicuspid valve. Notice there's a flap here and here that's been cut away. This one's been cut away. That's just two flaps. The bicuspid looks like this, and it kind of buzzes like a pair of lips. It can be, the 2 flaps can be pushed open and then they slam shut. Okay? So again, tri means 3, bi means 2. Those are your atrioventricular valves. Now we're going to talk about the semilunar valves. Semilunar valves are connected to the ventricles, and they control the flow of blood leaving each ventricle. So remember, you've got your right ventricle here that's sending blood to the lungs. It's going to be sending blood out through this valve that is separating the ventricle from the pulmonary artery. That's called the pulmonary valve. Nice and easy to remember. It's the valve that regulates blood flow to the lungs, pulmonary. Then, on the left side, you have the aortic valve. Now in this diagram, you can't really see it. Here's the aorta. Here's the path of blood going to the aorta. See this little dot? It's pointing to a valve right in here. Looks just like this one, but it's separating your left ventricle from your aorta. Okay? We call this the aortic valve. Regulates blood flow to the aorta. All right. Now, here's something to think about. These valves are made of fairly thin material. Okay? It's not very rigid, not very strong, and yet the blood that's flowing through the heart is under pressure, a lot of pressure. These valves are meant to prevent backflow. So when the ventricle squeezes, you want all the blood that's inside it to go in one direction, up and into the lungs. You don't want some of that blood flowing backwards back into the atrium. That wouldn't be very efficient. Every time your heart beats, half the blood goes backwards, half of it goes forwards. You're not going to make any progress. You want blood to flow through your heart in one direction. From the, see now I got to stop and get this straight. From the right atrium to the right ventricle, to the lungs, to the left atrium, left ventricle, back out to the body. Okay? One direction so that the blood picks up oxygen and then is able to deliver it. If these valves fail, if they break, blood is going to move backwards through the system and your heart's probably going to stop working. So how do you get these really soft structures to maintain their integrity and to slam shut and stay shut when the ventricle squeezes and puts them under pressure? Well, you connect them to muscles which pull down on the valve and help it stay shut. Okay? So here's your valve, or here's one flap of your valve, okay? This thing right here. And then check these strings out. Okay? These look like anchors, because that's what they are. They're called chordae tendineae. Think of cords and tendons, long, thin pieces of things that connect or anchor materials in place. Chordae tendineae anchor your valve flaps to a special muscle called a papillary muscle. The papillary muscle engages and helps to pull and keep the valve shut and in place when the ventricle is pumping. It also helps to prevent something called valve inversion. Imagine the pressure in this ventricle increasing as it compresses it's going to put a lot of pressure up on this valve, and it could cause the valve to, instead of just shutting like this, you could cause it to blow out and flip inside out, like when your umbrella flips inside out. It looks very similar when a valve inverts, and it's not a good thing. Okay, it means regulation between your atrium and your ventricle is no longer going to function. Okay. So the papillary muscles and the chordae tendineae help to prevent that. And that's it for this lecture. I'll catch you on the next one, where we'll talk more about these blood vessels.